Again, thanks everyone for participating and for supporting this kind of very unique and very important scientific enterprise that we are starting. And my lab is generally studying mucosal immunology, gut immunology, and I'm going to show today one of the interests of the lab, which is neuroimmune communications in the intestine. These guys in the picture are the people in the lab that have helped in these studies. But the important point I would like to make here is the collaborators that actually most of collaborators that we have in the lab in this topic now are part of FASI. And definitely this was initiated by joining this initiative. So again, thanks you guys for supporting it. One of the topics that actually we decided to cover today is neuroimmune interactions in the perspective of gut-brain communication. So I think everyone that is watching this, probably following main press, have followed in the past years the microbiota studies that showing that microbiome in the gut can influence every aspect of human biology. But particularly in the gut brain, of course, this is a major impact. So for example, in the perspective of the neurons, and I'm going to show some images of neurons that are in the gut, there is a large influence of the microbiota as a whole in the activity, in the number of these neurons that are located inside the intestine. But you also have an impact of this gut microbiome in neurons located outside the gut, particularly in the brain. And you probably heard about the influence of microbiota in depression, anxiety, in neurodegenerative diseases, including Parkinson's, Alzheimer's, as well as in autism spectrum disorders. So there's a large impact. A lot of this analysis still correlative, correlational studies, but there is also mechanistic studies showing that whatever is in the gut in terms of bacteria species may influence activity and the processes that are happening in the brain and influence the outcome of several of these diseases. So I thought it would be interesting to show you guys an image of an intestine. This is of a mouse, but in humans it's very similar. This I'm showing the ileum, so the small intestine, and just showing the neurons that are located in the ileum. You can see the process here. This is a 3D image. You can see the process that are both of neurons located within the intestine. This is what people call the enteric nervous system, what some people call the second brain. I'm sure for many people it's actually the first brain. But you also see here the processes that are leaving the intestine and going outside the intestine. These are neurons that are connected with the intestine, but not located necessarily within the gut. And just as a kind of overview of these neurons, you have not only in the intestine, the neurons that are part of the enteric nervous system, but you have processes that are involved in the sensing what is happening in the intestine and communicating these processes, this stimulation, or whatever stimuli that is taking place in the intestine to the brain. The brain, in contrast, can also regulate backwards what is happening in the intestine. So this is communication that is known to take place. What is not clear is how the gut can communicate with the brain and what is the influence, particularly in the gut microbiota and other stimuli that is happening in the gut, including allergic responses in behavior and also CNS, central nervous system processes. So that's one of the questions that the lab is interested in resolving. One of the main interests that we have is on these neurons that are secreting norepinephrine, noradrenaline, or adrenaline within the gut. This is a major impact both for immunity and for physiology of the intestine. And here you can see just this kind of neuron, which is called the sympathetic neurons, the part of the sympathetic arm of the nervous system. And I'm showing this because when we compared mice that lack the microbiota, the microbiome, or mice that were treated with antibiotic, what we found, and you can see here just the staining of a marker of activation in these neurons, that mice that lack microbiota show a spontaneous activation of these neurons that are secreting norepinephrine, the sympathetic neurons. And we wonder, because these neurons are located outside the gut, even though the processes are extending all the way to the intestine, we wonder how the microbiome located in the gut, it's influencing the activation of this whole chain of neurons that are located outside the intestine. So as a background on this, the sympathetic neurons that are innervated in the intestine participate in several processes of the gut, including blood flow, motility, so peristalsis, the way that the intestine moves the contents from proximal to distal intestine, fluid balance, and importantly, also it's known to influence immunity and allergic response. So this is a set of neurons that is crucial for several processes of the intestine. And again, it's a long story. I'm not going to spend too much time in this long story, but showing the summary, what we found that the neurons, the microbiota that is secreting metabolites in the gut, it's communicating and is sensed by specific neurons, sensory neurons, that communicate with specific regions of the brain. And these specific regions of the brain are then turning and regulating the activation of these sympathetic neurons located in the sympathetic ganglia outside the intestine. And we found that by using a variety of tools that actually many investigators in FACE have helped us, but basically are neuroscience tools. I'm going to show here one image of neurons in the brain that are indirectly via neuronal processes connected to the gut. So we found these neurons in the brain that are communicating directly via neuronal processes, neuronal synapses to the intestine. And these are the neurons labeled here. You can see in different regions of the brain. This is the whole brain that you clear and you visualize these communicating neurons. And what we did was, by modulating specifically the activation of these brain neurons that are communicating with the intestine, we were able to modulate peristalsis of the gut, which is one of the main, as I showed before, one of the main readouts or functions of these sympathetic neurons. So basically, by using a combination of microbiology, immunology, and neuroscience tools, we're able to identify neurons in the brain that are sensed via communication with other neurons, what is happening in the intestinal tissue, particularly the influence of the microbiota in the intestinal tissue. So basically, this is a summary of what we did. But in terms of going forward, what I would like to point is that many investigators, particularly in the case here, Rosa Medzitov, one of our main collaborators, and Esther Floschheim, also in FACE, we would like to characterize how these allergic responses that are initiated in the intestine are also communicating with the brain to modulate behavior in several physiological functions that depend on brain activity. So we want to know, for example, if the secret that we found that is sensing microbiome or microbial metabolites is also utilized in responses to food allergy. We also would like to define the neuroimmune partners that are participating either in the sensing aspect of this, meaning how immune cells can influence the sensory arm of these neurons, as well as the output, the end result of this communication. For example, whether neurons that are projecting and releasing neuropeptides in the intestine can influence the activity of immune cells. So this bidirectional interaction is part of some of our future studies. And again, I'm happy to answer questions at the end here, but I'm gonna go back to Chris. I know that we are slightly behind the time, but then again, thanks everyone for the attention on this.